I'm here today. So I grew up in Kansas City, Missouri, and I studied biochemistry at University of Missouri. And then after college, I decided to continue studying biochemistry and sort of working, like Dr. Lawson, sort of working in a lab and doing experiments like you would normally see like on TV with a white lab coat and all that kind of stuff. And I kind of figured out that I really wanted to do computational work and work with computers and do data analysis. And so at the time, Dr. Pevsner had an opening in his lab, and they happened to do human genetics research, which I was also really interested in. And so I'll talk a little bit about that and how it relates to Sturge-Weber syndrome. And you can see over on the right-hand side, I went through my photo library to see if I had any pictures of Jonathan. And that was the only picture that I had, was Jonathan sitting in the corner of his lab at his little computer and doing some work. And then the only photo I had of myself was my ID card. So I stuck that in there as well. So after being in Jonathan's lab for a number of years, I graduated and then stayed. I met my wife at Hopkins, so I also stayed there, and we did some extra work. And I studied oncology, so I switched over to studying cancer biology. And now I work as a cancer biology researcher. I do computational data analysis, and I still do human genetics work, but it's more focused on finding cancer drugs, and I work for Novartis. And then, like Julia said, I took over this year for Jonathan as the Chief Scientific Officer for the foundation, and I'm very, very excited to be able to meet everybody and help us sort of progress and understand more about Sturge-Weber syndrome and really try to get us closer to the goal of having some type of beneficial drug, therapeutic, that might help not just control sort of symptoms and improve quality of life because of symptoms like Dr. Sandhar was just explaining, but really understand Sturge-Weber syndrome as a genetic disease so that we can actually come up with solutions for the root cause of those symptoms. Okay, so really briefly, I just wanted to explain what Jonathan and I did in his lab. So back in 2013, we had a collaboration, so we were working with other researchers to try to understand, what causes Sturge-Weber syndrome. And for that to happen, we really had to have involvement from patients who were actually donating human tissue. So skin from Port-Wine stain, affected brain samples from hemispherectomy surgeries that were graciously donated, and then also blood from just sort of like normal blood. And around 2013, we had access to very new, exciting technologies in human genetics. So almost all of human genetics that we understood before maybe 2008 was understood in a very, very, a very tedious way. We found a group of people who all had a very similar, what we'd call a phenotype, but that's something about them that we can see. So brown hair, green eyes, that kind of thing. And then we'd go find those people, and then almost like looking through a 1,000-piece jigsaw puzzle without putting it together, we'd go try and find the one exact piece in their puzzle that they shared. And you can imagine that this work is very tedious, and it was very difficult and very time-consuming. And around 2013, we actually started to have access to things called next-generation DNA sequencing instruments. And you can imagine this like taking all of those puzzle pieces, being able to understand all of them almost at the same time. And people started building tools to start putting these puzzle pieces together in ways that made sense so that we could understand genetic diseases more quickly and easily. And so I'm explaining this because part of the work that we did in 2013 in Jonathan's lab was actually just lucky. We kind of were around at the right time to make these discoveries. So we had the DNA from affected individuals with Sturge-Weber syndrome, and we sequenced their genome. And then we compared the genome from the port-wine stained skin and compared that to the normal blood. And what this allowed us to do is understand, you know, if I go back to that puzzle piece analogy, understand which puzzle pieces these individuals shared in common. And from that, we could start to say, well, we think that those pieces of DNA are actually what's causing Sturge-Weber syndrome. And so we published a paper on this. We had 3 individuals donate their skin and brain tissue to the Maryland Brain and Tissue Bank. And after we sequenced those 3 individuals' genomes, we went back and found 49 other patients. And from these 49 other patients, we were able to go in, now that we felt we understood that this GNAQ gene was causing Sturge-Weber syndrome, and we were able to ask if that GNAQ mutation was found in all of these 49 patients. And so the short answer is yes. Most of these patients, about 80 to 90% of brain and skin samples from Sturge-Weber patients had the GNAQ mutation that we found. And so since then, other researchers have tried to identify mutations in those remaining maybe 10% of Sturge-Weber patients where they did not have the GNAQ mutation. And it turns out that the other genes that that might explain these remaining Sturge-Weber cases are also very closely related to GNAQ function. So I was just extremely grateful in Jonathan's lab that we had these generous donations of tissue from families and patients. It's incredibly useful for basic science, and where basic science is just understanding how things work in your human body, to be able to have, sorry, I got a little feedback there, to be able to have these tissue donations that really enables the science. And then we had funding through the Sturge-Weber Foundation, which was also incredible. And then, like I was saying previously, we had good timing and luck. So this was 2013, and now it's 2021. So we've got sort of 8 years that's gone by since we sort of understood the genetics of Sturge-Weber syndrome. And I don't want to make too big of a deal about understanding that the mutation in GNAQ causes Sturge-Weber syndrome. I think it was really fantastic that we can make this discovery, but just understanding which gene causes a syndrome doesn't necessarily help you understand how to develop better treatments and how the syndrome actually develops in affected individuals. And so we still have a lot of work to do. So one thing that we really have to do as scientists is build what are called animal models of Sturge-Weber syndrome. So the first one here that I list is actually a little bit weird, and people typically don't understand why we would make fish models to study human disease. But what I'm showing on the right-hand image here is there's actually a cancer called uveal melanoma. It's a cancer of the eye, and it actually shares the same mutation that causes Sturge-Weber syndrome, which is a little bit confusing. Sturge-Weber syndrome is not a cancer. And so the reason why you can have one mutation that causes 2 different phenotypes, syndromes, is that the mutation can happen in different cells in your body. And zebrafish are transparent, and you can see all of the different cells inside the zebrafish. So one thing that we can do is make mutations. Mutations sound bad, but in this case, we want to actually make mutations that can cause something that looks like Sturge-Weber syndrome or something that looks like this uveal melanoma. And that's one way that we can understand how the syndrome can develop in humans. The other thing that we're working toward is making mouse models. So mice seem a lot closer to humans than zebrafish, and actually genetically that's true. Sometimes they're very good surrogates for understanding how diseases progress. And so we're working with a number of groups toward creating these genetic mouse models. And the reason why these are important for basic science research is because we need to understand if we think we have a drug or a type of therapeutic that can really help with the symptoms of Sturge-Weber syndrome, then the first thing the FDA wants you to do is to test the effectiveness and safety in an animal model, and mice are a very good starting point for this. And then the other thing that we really hope to do is translate some of the existing research. In this case, I'm listing uveal melanoma. So this is another, this is a cancer that shares, again, like I said, the same genetic cause as Sturge-Weber syndrome. And there's starting to be some very good clinical trials for uveal melanoma, for therapeutics that directly target GNAQ, or drugs that target things related to GNAQ. And these are all therapeutics that we could imagine could be useful to understand for Sturge-Weber. But again, the first thing that we have to do is understand the cause and development of Sturge-Weber syndrome in a little bit more detail. But I think that there are various biotechs, so working for a large pharmaceutical company now, and working in my day job on uveal melanoma, I'd really like to leverage the work that's going on in pharma to try to translate some of the work and the money that goes into the investment that goes into treating uveal melanoma, and see if that can help us out as well as a patient population. Okay, so what are we doing as a foundation, right? I said that for my work with Dr. Pevsner, we had a foundation grant that really enabled the genetic research for Sturge-Weber syndrome. And we're continuing that as a foundation now. So last year, the foundation issued a grant to Dr. Naeem Issa, and his work involves taking drugs that are already approved for other purposes and trying to understand if we can repurpose those drugs to treat Sturge-Weber syndrome. And the main reason that we want to do this type of work is that it's very difficult and very expensive to develop a new drug and get that approved for use in humans by the FDA. And it's also really difficult to understand the safety of a drug that you might want to try. And so when all of that work has already been done by somebody else for a different purpose, then it would be fantastic if we can actually find a therapeutic that we already understand a little bit about the safety and see if it's effective in Sturge-Weber syndrome. So Dr. Issa actually has some very interesting preliminary results, and it's been great to be able to help that research along. And we also issued a grant last year to Dr. Arbisser. And, in putting together this presentation, I was thinking about these animal models that we want to create. But for the basic science research, where basic science is just understanding how your cells work, how does the GNAQ mutation make your cells in your body different? And we think that Sturge-Weber syndrome is a neurovascular disorder, and so it affects blood vessels, so those are vascular cells, and may also directly affect a neural cell type, or maybe the vascular cells are actually causing all of the neurology. But Dr. Arbiter's group is trying to put together even more basic models, not just a mouse, but actually just like vascular cells. So just the cells that might be involved in the Sturge-Weber syndrome development. He's putting together these mutant cells. Again, mutant sounds like a bad thing, but in this case, it's actually a good thing, because we can then use these cells to study the effects of different drugs and different therapeutics that we could imagine might have a benefit for patients with Sturge-Weber syndrome. So these are all different tools that we need to develop so that we can eventually get enough evidence to test some therapeutics someday in a clinical trial with humans. And we want to be very sure that those are safe and effective. So I don't want to go on for very long here. I just wanted to give kind of a flavor. And certainly I don't understand coming into this role. I've only been CSO for a little over a month. But I wanted to give a little flavor of the type of work that we're trying to do as a foundation through our partnerships with scientists and clinicians to try to understand how to push the science of Sturge-Weber forward and help patients. So what can we do to help, right? We can continue to build our relationships through the Sturge-Weber Foundation Clinical Care Network. So these are all the clinicians that you families will be very familiar with, because you're actually seeing them and building your own relationships for managing Sturge-Weber symptoms. And these clinicians should be able to help us build a better clinical data resource. So by that, I mean, we want to understand when families and patients are treating symptoms like seizures, what medications are they using, how often are they using them, and what does treatment look like? And that can actually help us understand a little bit more about the progression of Sturge-Weber syndrome. And we can also link those data eventually with information like what we generated in Dr. Pevsner's lab. Like we did whole genome sequencing, but there are other different types of big scientific research that we can do with these generous patient donations of skin and brain samples. And when those opportunities arise, it's great to be able to understand more about the patients that made those donations. And then also joining the Sturge-Weber Foundation International Registry. I think that's a great idea. And that's just to help us capture information about, you know, what it's like from a patient perspective, what your symptoms are, things that you might not necessarily be capturing and treating with your physician relationships. But we have, I think, a bunch of different questions in that registry that can maybe help us find, you know, is there a link between hormone development through puberty onto adulthood, and try to understand the syndrome in a more comprehensive way. And if you have any questions, please feel free to reach out to me, you know, over email. I'm available. And I think we have time for any questions right now. This is my last slide.